Welcome to your fifth video on cell biology, and in this video we're looking at unit 1.3, which is all about membrane structure. Okay, so when we're looking at membrane structure, we're looking at our plasma membranes that surround all of our cells. And we've already said that the role of that is to contain the cell together, but to also allow certain substances that were required within the cell to pass across, but also to manage what passes out of the cell, to keep that cellular equilibrium. Okay, so all cells surrounded by this membrane, and it's to control the movement of substances in and out of our cell. Now, on our eukaryotic cells, our organelles are also contained within plasma membranes, and this allows each organelle to have a controlled environment as well. So what is our plasma membrane? And we've got a picture of a plasma membrane there. So it's composed of what we call our phospholipid bilayer, and we're going to look at this further. And this phospholipid bilayer is embedded with proteins which have a certain roles. Now we call this idea and this concept of these phospholipid bilayer with these proteins embedded, we call it the fluid mosaic model. Now, when we think about that term, fluid mosaic model, think about what a mosaic is. It's a representation made up of different shapes as such. So you'll see, or shapes and colours, you'll see on the left hand side there's a picture of a mosaic. And if you look on the right hand side as our image of our fluid mosaic plasma membrane, you'll see where that idea kind of comes from, that mixture of different shapes and patterns in our membrane. Okay, so what is it about this fluid mosaic model? Well, it was proposed in the 1970s by Singer and Nicholson, and it was after a lot of research that they'd put into intercellular biology and looking at what keeps that cell all together. It is our current accepted theory of membrane structure. Remember, I said you're the biologist of the future. This could change, but this is the current accepted theory. Alrighty, so these membranes are quite complicated. Okay, so in the past, you've drawn them just as that little membrane around the cell. They are actually quite complicated in terms of their construction, and they're absolutely fascinating. So we're going to start off by looking at different components of the membrane, and our first one are going to be the phospholipids. These are really cool. Okay, so we've got this phospholipid bilayer. Okay, so if we look back at our fluid mosaic picture, this is our phospholipid bilayer here. Okay. So we've got these two layers of these little things with this kind of head and two little, literally kind of legs on it, okay? And the way this forms is these molecules have a hydrophilic head, so a water-loving head, and hydrophobic tails, so water-hating tails. Okay, so think phobic, scared, hating. All right, so it's made up, our phospholipid bilayer is made up of one type of molecule, and it's our phospholipid. Now, they're what we call amphipathic, okay? This means they have regions that are both hydrophobic and hydrophilic. So they have regions that like water and regions that dislike water. Okay, so let's look a bit closer at our phospholipids here. We've got that hydrophilic head, so that's attracted to water, and our hydrophobic tail, repelled by water. So the words hydrophilic, attracted to water, hydrophobic, repelled by water, and then our amphipathic is substances that have parts that are both hydrophilic and hydrophobic. Alrighty, so we've got those terms, we've got that idea in our head, but what is our phospholipid actually made up of? So we've got a phosphate, we've got glycerol, and we've got two fatty acid towels, so our hydrocarbon towels. The phosphate and the glycerol form what we call that hydrophilic head, and the fatty acid towels, they're our hydrophobic towels. And because we've got the hydrophilic and hydrophobic, our molecule is amphithetic. So how does the bilayer actually form? So in a watery environment, the phospholipids naturally will arrange themselves to have their hydrophobic towels inside, protected from the watery environment as such. So that's why they align in the bilayer. Their hydrophobic heads line up to protect their hydrophilic towels in between. So both in the cell and out the cell, both watery environments, that's why they align in this manner. Okay, so we've looked at the phospholipids. We've looked at that phospholipid bilayer. Okay, so that's kind of the big construction. But then embedded in that are other components. And first, we're going to look at our proteins that are embedded in our plasma membranes. So there's various proteins and they have various roles. Okay, so here we can see our channel protein. We've got another protein here. There's another protein over there. Okay, so each of them have different roles. So they're for different reasons. So some of their functions, they transport materials across the membrane. They're involved in cell-to-cell communications, and they can be hormone binding sites as well. Now, our proteins can be described in two ways. They can be integral proteins, and generally these have part of them that are hydrophobic, so they're within the cell, and these often go transmembrane, so across the entire membrane. Okay. We also have what we call peripheral proteins, and these are the ones that are found on the edge of the plasma membrane. Okay, these are generally hydrophilic, so they're found on that hydrophilic surface. Some can have hydrocarbon chains attached, and we quite often call them a glycoprotein. So some of the roles of these, our integral proteins, they can be immobilized enzymes ready for metabolic reactions. We can have channels for passive transport, which goes through the protein. So you see there, passive transport can occur through there. And we also have pumps for active transport. Now we're going to look further at transport in and out of the cell in a unit further on, but this is just introducing that area. So this allows diffusion to occur into the protein, so passive, not requiring energy process, and active transport requires energy to move substances into the cells. So there's three different roles of our integral proteins, those proteins that go right through the cell, and then some roles of our peripheral proteins. We've got these hormone binding sites, our cell-to-cell communication, and some cell adhesion happening. Okay, so another component that we're going to look at is the cholesterol. And you'll see cholesterol on our diagram here as represented as the little yellow hexagons. So these are embedded within the fatty acid towels. Okay, so within those hydrophilic towels. Basically, our cholesterol maintains the fluidity of the membrane. Okay, so remember it's that plasma, and the cholesterol allows that fluidness of the plasma to maintain. In mammalian cell membranes, the cholesterol reduces the membrane fluidity, so keeps it at a constant level for where it needs to be, and it's also involved with the permeability for some solutes to pass across the membrane. So remembering that this fluid mosaic model was proposed in the 70s by Singer and Nicholson. Okay, so they were who proposed it, and this is our accepted theory. So where does that bring us to? If in the 1970s this model was proposed, and now there's further developments being looked at in terms of cell membranes. As I said, it's ongoing research. This is your future, guys. However, what occurred before the 1970s? And that's what we're going to look at now. So part of your course in terms of your nature of science is using models as representations of the real world. So we're looking at this model of the fluid mosaic membrane. Okay, another part of your nature of science is that falsification of theories. So how you can say that a theory is incorrect, okay, or has been surpassed by another. So how has the fluid mosaic model falsified a previous model? And we'll look at the Davison and Daniela model. Now, we're also looking at some skills here. We're looking at the analysis of evidence from electron microscopy that led to the proposal of Davison and Daniela, that model. Okay, and we're also looking at the falsification of that model that then led to our Singer and Nicholson model. Okay, so back in the 1930s, Davison and Danieli proposed a model that explains membrane structure. Okay, and this is their model here, the sandwich model. They proposed that the membrane composed of 2 layers of proteins and a phospholipid layer in between. They called this the sandwich model, and it explained how very thin membranes are an effective barrier to the movement of some substances. This was supported when in the 1950s, high-powered electron micrographs showed a railroad track appearance between 2 dark lines, the proteins, and a lighter band, the phospholipids. So it was supported by the technology that they had. The model was known as the Davison-Danieli model, so that sandwich model. So when did that get proved incorrect? What's the falsification? So falsification of theories occurs when one theory is superseded by another. So how did that sandwich model get superseded by our current fluid mosaic model? So in the 1950s and 60s, experimental evidence accumulated and led to the falsification of the Davison-Danieli model of membranes. So freeze-etched electron micrographs, okay, this is what actually led to the falsification of that. Technology developed and allowed this freeze etching to occur. These methods involved rapid freezing and then fracturing of the membrane, which resulted in fractures occurring along lines of weakness, including the center of the membrane. Globular structures scattered throughout were identified as our transmembrane proteins. Proteins were found to be varied in shape and size and not consistent with the one continuous layer that had been proposed in our sandwich model. Then, after our freeze etching, we also had fluorescent antibody tagging. So this also proves the falsification of this model. So red or green fluorescent markers were attached to antibodies that bind to membrane proteins. The membranes of some cells were tagged red, while other cells were tagged green. The cells were fused together, and after 40 minutes, the red and green markers were mixed throughout the membrane of the fused cell. This showed that the proteins were free to move within the membrane and not fixed just to the outer layer. So if we then compare the two models by structure, by a diagram here, you can see how it's developed on. With the initial Davison and Danieli model, those proteins were just along the top and along the bottom. Still both have the phospholipid bilayer. However, the falsification of that model has shown that those proteins are not just along the top and the bottom. They actually are through the biphospholipid layer, they are on top, they might be below, but they don't just sit along the top and the bottom. And we've finished that section, finished unit 1.3. And I'll see you soon in our next video. Thanks for watching.